Radio Khanchi 90.4 FM, Hum Sabka Radio. Myself, Malay Bharti, Assistant Professor, Postgraduate Department of Botany, Dronda College, Ranchi University. Dear students, in this session, I will be talking about immunity response, that is antigen-antibody immunity response, which is a part of syllabus of botany at PG level, especially semester 4. Dear students, there are three important terms which we are taking up right now, that is immunity, antigen and antibody. So before starting, let us see what antigens are. Actually, antigen is a substance which, when introduced into a body, evokes an immune response to produce a specific antibody with which it reacts specifically. So this is the antigen. Now, antibody is another very important term. What antibodies are? These are substances which are formed in serum and tissue liquids in response to the antigen and reacts with that antigen specifically on some absorbable manner. Chemically, these antibodies are globulins, hence they are termed as immunoglobulin. They contain 20 to 25% of the total serum proteins and are mainly synthesized by plasma cells. So there are different classes of immunoglobulins, or you can say the type, that is IG, that is immunoglobulin. So IgG, IgA, IgM, IgE and IgD. So these are the different classes of immunoglobulin. Generally IgG, which protects the body fluid, IgA, it protects the body surface, IgM, it protects the blood stream, IgE, it mediates rhiogenic hypersensitivity. So these are some types of immunoglobulin and their main activities. Now, there are two important terms, dear student. One is epitope and another is paratope. So what epitopes are? Actually, epitope is very smallest unit of antigenicity. It is present on the reacting site, that is actually the reacting site of antigen, epitope. That is the smallest unit of antigenicity. And paratope, it is constituting or combining site on the antibody molecule which is corresponding to the epitope, and hence it is called paratope. So paratope and epitope, they are very much complementary to each other. One epitope on the antigen and another paratope on the antibody. Now let us concentrate the main terminology, that is immunity, main term, that is immunity. What immunity is? Nowadays we are very much listening that immunity is to be developed to save ourselves from different viral infection and all. So let us understand what immunity is. So immunity, actually this term is derived from immunitas, that is a Latin word which means for exemption from civic duties or paying taxes. So the term immunity is defined as a resistance exhibited by the host against any foreign antigen, including microorganism. So this is actually the defense factor of the body against any microorganism in general sense. So immunity, it may be different types, it may be innate, it may be acquired, it may be miscellaneous immunity. So three important immunity, it may be innate, it may be acquired, it may be miscellaneous. Innate immunity, again, it is specified into species innate immunity, racial innate immunity, and individual innate immunity. Likewise, acquired immunity also it is divided into two, that is active immunity and passive immunity, and both are further divided under natural and artificial immunity. Likewise, miscellaneous immunity also it is divided into adaptive immunity, local immunity, and herd immunity. Now, let us try to understand what innate immunity is. Actually, this innate immunity have been discovered by Metchnikoff in 1908. It is directly response to the pathogens, direct response to pathogens, and it is conserved microbial products, or it helps in the conserving microbial products, not antigen anti-specific. This is not antigen anti-specific. So adaptive immunity, again, it is discovered by Ehrlich in 1908. So in 1908, adaptive immunity was discovered by Ehrlich, which is actually specific antibodies, and it causes stimulation of T cells and B cells. They are variable antigens. So T cells stimulation requires antigen presentation by antigen preserving cells, that is APCs, antigen preserving cells. Now, recognition of T cell receptors for their target does not necessarily lead to cell killing. So now, in order for T cell to do their job, helping B cells respond by making antibodies and also by helping to kill virus infected cells and other types of pathogen. So actually they themselves requires a significant amount of help which is provided. Now let us concentrate antigen antibody reaction or response. In this process, the antigen and antibodies combine specifically with each other. This interaction between them is called antigen-antibody reaction. It may be abbreviated as AG antigen, AB antibody reaction. So the first correct description of antigen-antibody reaction was given by Richard J. Goldenberg, very famous pathologist Richard J. Goldenberg at the University of Wisconsin, 1952. So these reactions form the basic form of detection of infection diseases or infectious diseases causing agent and some non-specific antigens like enzymes. The reaction between AG antigen and antibody occurs in three stages. First, the primary stage is the reaction involves formation of AG-AB complex, that is antigen-antibody complex. It occurs in the primary stage. Now, secondary stages which leads to visible event like precipitation and agglutination. So in the second stage, when complex AG-AB complex are formed, then precipitation occurs and agglutination occurs, which are very much visible and clinically visible during different tests. Third stage is tertiary test. Actually, it is destruction of antigens or its neutralization. So here in this third stage, the antigens are destroyed or it may be neutralized. Dear students, one thing which is very important, that this type of specific reactivity induced in a host by an antigen stimulus is known as immune response. It helps in protection against invading microorganisms. It may lead to consequences which may be either beneficial, indifferent, or it may be injurious. The immune response can be of two types, that is humoral mediated immunity, that is HMI, and another is cell mediated immunity, that is CMI. So usually developed together, but either may be predominant, exclusive, or sometimes act together, or sometimes in opposite. So let us see what HMI, that is humoral mediated immunity, are. Actually, humoral mediated immunity, it is antibody mediated immunity. So antibody here produced from plasma cell and that present in blood and other body fluid, that is humoral body fluid. It provides defense against most bacterial, also participates in pathogenesis of type 1, 2 and type 3 hypersensitivity reaction and autoimmune disorder. Now the other one, that is CMI, cell mediated immunity. So this cell mediated immunity is very specific immune response that does not involve antibody. It protects against fungi, virus and facultative intracellular bacterial pathogen. There is rejection of homografts and graft versus host reaction. So it is also providing immunological surveillance and immunity against cancer. It mediates pathogenesis of delayed type 4 hypersensitivity reaction and certain autoimmune disorder. Now, let us try to understand that what happens in HMI, that is humoral mediated immunity. Actually here there is production of antibodies in 4 phases. One lag phase, log phase, plateau phase and decline phase in HMI. So in lag phase, actually entry of pathogen and its fate in tissue is decided. Log phase, there is a steady rise in situ the titer of antibody. Plateau phase, there is equilibrium stage occurs between antibody synthesis and catabolism. And in decline phase, actually this phase is catabolism which exceeds production and thus titer value falls. Now, here in HMI, that is humoral mediated immunity, first antigen enters into the body, then antigen presenting cell, that is APC, gets activated. As soon as this APC gets activated, it produces macrophages and dendritic cells is stimulated. Now APC, that is antigen presenting cell, binds with major histocompatibility complex 2, MHC2, major histocompatibility complex 2. Then immature T cell binds with earlier formed complex with the help of T cells receptor, that is TCR. Thereafter, whole complex produce signal for activation of CD4 cells, then maturation of T helper cells, that is TH cell, takes place. So in next stage, there is formation of IL, that is interleukin 2, interleukin 4 and interleukin 5 and 6 occurs. So B cells maturation occurs and subsequently releases the plasma cells. So plasma cells forms antibodies, some of the plasma cell forms memory cells. So here it is known as polyclonal antibody. In this case, there is continued production of antibody by cloning of plasma cells. And in other cases, which may be known as monoclonal antibody, so according to Kohler and Milstein, they were the Nobel Prize winner in 1984, a single antibody forming cell or clone produced antibody against single antigen, but usually antibodies are polyclonal. Now, let us have an idea of cell-mediated immunity, CMI. What happens in this case? Actually, a specific immune response that do not involve antibody. So here antibody is not involved in CMI. It participates in following immunological function like delayed hypersensitivity, then immune infection caused by obligate and facultative intracellular parasites like bacteria, fungi, parasites and virus. Bacteria, which is causing tuberculosis, leprosy and brucellosis. Fungi, for example, causing coccidioidosis and blastomycosis. Parasites causes leishmaniasis and trypanosomiasis. Viruses causing measles, mumps, etc. So these are the certain examples. So there is immunological surveillance and immunity against malignancy actually. So pathogenesis of autoimmune disorder like thyroiditis and encephalomyelitis. So these are the pathogenesis of autoimmune disorder. Now actually induction of cell-mediated immunity, how it takes place. So when antigen enters, this antigen presenting cell APC gets activated and it produces macrophage and dendrite cells, or it may stimulate this macrophage and dendrite cell. Then APC, antigen presenting cell, binds with MHC1, that is major histocompatibility complex 1. Thereafter immature T cell binds with earlier formed complex with the help of T receptor cell, that is TCR. Now, the whole complex inside the formation of CD4 helper and CD8 cells, these cells are cytotoxic cells. So, former CD4 cell releases the lymphokines which activates macrophage and eats away intracellular parasites. CD8 cell recognizes antigen on surface of virus, infected cell, tumor cells, allograft cells with MHC1, major histocompatibility complex 1, and secretes lymphokines and finally destroy the target cell. Now cytokines, what cytokines are? Actually they are the signaling protein and glycoproteins that are used extremely in cellular communication. So it regulates immunological inflammation or inflammatory and reparatory host responses. So these cytokines and glycoproteins plays very important role and it acts like hormone and neurotransmitter. It's grouped into 5 classes, that is interleukin, IL-1 to 13 number, IL-1 to 13, colony stimulating factor CSF, tumor necrosis factor TNF, interferon INF and others like transforming growth factor TGF and leukemia inhibitory factor LIF. So dear students, now let me summarize as a whole that activation of immune response typically begins when a pathogen enters the body. So macrophages that encounters the pathogen ingest process and display the antigens fragment under the cell surface. So these macrophages with antigen fragments laid on the surface are called antigen presenting cell APC, and antigen presenting macrophage in its action helping T helper cells and can recognize the same antigen. So during the interaction, the macrophage releases a chemical alarm signal called interleukin 1, that is interleukin 1, which stimulates the T helper cell to secrete interleukin 2. So interleukin 2 causes proliferation of certain cytotoxic cell, T cell and B cells. These immune response from this point follows two poles, one using cytotoxic T cell and another using cytotoxic B cell. Normal cells of the body that become infected can also digest some of the pathogen and display antigen fragments onto their cell surface. So body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular type of antigen. Now, here cytotoxic T cells that are capable of recognizing the antigen displayed onto the surface of infected cell, it binds so the infected cell and produces chemicals that kill the infected cells. Thereafter, infected cells results in the destruction of pathogens. These cells also common in millions of different types and each able to recognize the particular antigen. So when B cells becomes activated by T helper cells, they differentiate into plasma cell. These plasma cell becomes antibiotic producing factories, which causes flooding of the blood stream with antibodies, and they can bind the antigen involved in the infection. So antibodies bind to the antigen on the surface of the pathogen, marking them for destruction of macrophages. Some of these B cells do not turn into antibody factories but instead become memory B cell that may survive for several decades. Because of these memory B cells, the secondary immune response to the future infection by some same pathogen is swifter and stronger. So this powerful secondary immune response is what kills immunity to some diseases after you have them once or after you have been vaccinated. So dear friends, this is all about the immune responses, and you may get much more in the book on microbiology written by Anantharajan and Panikkar, and another book on Lippincott's Illustrated Review of Microbiology, 3rd edition, and another book that is written by Belgi. So I hope you have enjoyed this talk, and thank you for listening. Thank you very much.